record this conversation, and I will have a copy for Maurice Israel.
Sounds perfect.
Okay. Thanks a lot to come.
Not at all. Our pleasure.
Okay.
Would you like to just walk us through your experience, Mr. Tenenbaum? Would you like to tell us? I've read I looked at your website, looked at your videos, looked at your papers, but it'd be useful to hear from you what you're looking.
I did also 3 books where I explain everything, but I will try to be short to not waste our time. In 2018, I was diagnosed with cancer stage 4, Gleason group 5, Gleason 8 and 9. So 7, 8, and 9 to be precise. And Dr. Pizzolato, he was in another hospital, I forgot, Mount Sinai Hospital in this time. And all the oncologists I saw in France, in USA, in Spain, told me that I'm dead. Okay, 3 months to live. Okay, so nothing to do, enjoy the life. It was not my conception of life. And I start studying on PubMed, on Nature, on all this publication, the Nobel Prize. And when I understood that cancer is a metabolic disease, so I build my cancer myself. And I went to see Professor Thomas Seyfried. And this guy said since years that cancer is a metabolic disease. So I went to see him and I told him, I would like to be your guinea pig because what you are saying, I think it's true because I do what you say and I feel better. So he told me, record each day what you are doing. That's why I have 2,000 videos today. And to follow chronological, I don't know if we say it this way, what I did. And I knew a lot of professors, a lot of Nobel Prize. I searched everywhere, how can I survive? And finally I saw Professor Maurice Israel, who is, he was chief of one of CNRS in France during 15 years. It was a director. And this guy studied, after his retirement, he studied cancer since 20 years. And he was passionate about what I did, how I did, because what I did confirmed what he was thinking. So we worked together since 4 years. And I'm sure we find a way to kill cancer cells, because they are only one door to the way you can kill cancer cell without affecting the other cells. It's called SCOT for him. SCOT is OXCT1. It's an enzyme. And they are only one door a cancer cell can use. And this is this door. And I spoke about that with Thomas Seyfried, and Thomas Seyfried doesn't agree. He said it's the inhibition of mTOR and the inhibition of ROS. So I study and I think, and if the four of them are the solution, ROS, mTOR, OXCT1, and TIOLAs. Why TIOLAs? Because without TIOLAs, you cannot see OXCT1 because it's too fast. Then the step we need today, it's the Williamson dosage. We have to buy the Sigma. Sigma is a brand where we can buy everything. Enzyme, a purified enzyme of OXCT1. We have to separate that, put it in a freezer, and use little by little with different things inhibit SCOT. And to do that, we need a spectrophotometer continuous to register what happened. We need to test the absorbance, I don't know how you call that in English. And we need to find the best siderophore, because all the siderophore are probably inhibitors of SCOT. Why? Because it's acid hydroxamic. And we know, because FDA approved 2 medication against cancer who are some of derived, almost the same than acid hydroxamic. It's Pimozid and Litostat. And we did a publication on that. And we search, I had help, of course, we find in Spain the same thing than Litostat. It's called Uronefrex. And you can buy it very easily in Spain. And we study what are common in other products. What is common with acid hydroxamic. And we find the red algae, litotam, phycocyanin, and melatonin. And melatonin is well known to kill cancer cells. But we know that melatonin kills cancer cells, but we don't know why. We don't know what melatonin is doing, but we don't know why it's doing this. And Maurice Israel explained to me, it's because inhibition of OXCT1, SCOT. So today, the first step, what we have to do, it's to find the best inhibition of SCOT. We are thinking that it will be melatonin, because another hospital in France, Gustave Roussy, I don't know if you know them, are using until on research. They use 450 milligrams per day to kill cancer cells. But we don't follow this way, because I think that it's too much. There are no side effects on melatonin, so why not? But it's not enough. We need to use what we call the 5 essential, the 5 essential. And the 5 essential is other inhibition. The first one, epigallocatechin, the green tea, of course. The garlic, allicin, and the discovery of Maurice Israel in 2010, the metabolic treatment, it's acid lipoic and Garcinia cambogia. With this, all my cancer cells disappear. I just said to John that even in my PSMA, I have no cancer cells at all. It's brand new. I don't know if you saw it, but Maurice Israel tell me it's normal because cancer cell cannot use acetyl-CoA, and this way cannot do membrane, and this way cannot live. So I think it's really a very important discovery.
So, I appreciate you going through all of that with me, Mr. Tenenbaum. I wanted to just sort of, again, like I said, I think probably a ton more metabolism than I know. I just want to see if Dr. Belusic, I want to sort of get his clinical perspective. Then I'll tell you, some of my thoughts. I think you've raised some really interesting and key points. So I don't know, but I just want to sort of turn the floor over to Mario first.
Sure. Can you hear me?
Yeah.
Okay, nice to meet you, sir. So I'm the...
Hi, I say hi.
Hi. So I'm the prostate cancer medical oncologist. I'm not sure how much Dr. Rai introduced me, but I was trained at the National Cancer Institute in Bethesda, and I was working there for like 10 years, total, during the training, and then later as a faculty, immunotherapy and prostate cancer. I was also the fellowship program director, so I was in charge of training of junior, hematologists and oncologists. So, I have this great, idea that I learned from my patient prostate cancer, and I was studying metformin for prostate cancer, and based on your concept that this is metabolic disease, and there are lots of, interesting discovery, and that was, that idea was driven by my patients who then, talked to me. They took the medication, everything. And the other project I did as when I was before the NCI, I was in the Fox Chase in Philadelphia. I did work with exceptional responders, and those are patients not only with prostate cancer but also people with different type of cancers who had dramatic response on the treatment they received or whatever they did.
Metformin. You said metformin.
Yes.
I will tell you after. I will let you finish.
Yes.
We study this case too.
Yeah, so metformin, we did a study and showed that 40% of patients had a response to metformin alone, and Dr. Rai and I are working now on the next step and trying to understand what's going on, how this works. We know there was some negative studies in castration-resistant prostate cancer, but it seems that castration-sensitive prostate cancer is a different type of disease, and based on our work, it seems that some people are responding. I mean, lots of my people, that was a 28 patient study, and some people after it was only for 8 months, people decide to stay on metformin later, and they really had good response, and they avoided the hormonal therapy which was prescribed to them and offered them by their doctor. So I think completely, there is definitely something about metabolism, and I'm not telling them metformin is the key, is the right thing to do. That was just what I was studying, but there are many more things that we don't know. And I think studying the metabolism makes complete sense, and the impact on the cancer itself. I also like immunotherapy. I also think that immune system play a crucial role, and I think there's a huge interaction between the metabolism and immune system, because one of the main role of immune system is to fight a cancer, and we know that some people, they have defective immune system. Let's say people that have HIV or they're on immunosuppression or organ transplant, they have 20-fold increased risk of cancer, including prostate cancer. So there's definitely multiple things, and there are multiple mechanisms that are, playing a role. I'm happy to hear that you had such a great response to your intervention, and I think definitely it's worth studying and investigating, trying to understand this mechanism better.
So, Mr. Tenenbaum, tell us about your experience with metformin.
Okay. Dr. Laurent Schwartz, I don't know if you know him, did studies on metformin on mice. It was a great success. And we saw, like you, Dr. Mario, that it doesn't work with all the patients. And we discovered something very important, I think. You know, of course, Jason Fong, and he said that metformin take the sugar from your blood and hide it in your fat, in your fat cells. So when you do what propose Thomas Seyfried, it's ketogenic diet, you lose weight. And when you lose weight, all this sugar go back in your blood. I did it. I lost a lot of weight, but I had until 240 instead of 85, 90 in my blood. Then it's dangerous and it doesn't work for everybody. It can work if you don't lose weight. If you don't lose weight, the sugar stay in your fat and you have no problem. But if you want to kill cancer, you have to do a keto diet. Why? Because you don't eat sugar, carb, no carb at all. In the first phase, because we know that cancer cell use sugar to feed it, yeah. And glutamine, like Thomas Seyfried said. But in keto diet, we have what we call a paradox, because you have two ways: the ketones or the ketones are only signalytic. I'm not a biologist, I cannot explain that very clearly, but it's very easy to understand. If it stays like a signalization, it works. And if the ketones can be the nutrients of cancer, you die. That's the keto paradox. That's why the Chinese did several studies saying it doesn't work. Yes, because it doesn't work when the cancer cells can use ketones. And this Professor Thomas Seyfried does not agree with that. He said cancer ketones cannot ferment and cancer cell cannot use it. But the way, and I will give you a dibujo in Spain, a dessin, a drawing, yeah, from Maurice Israel. We explained very clearly that the way we know that cancer is smart, and he used another way, not fermentation. But this is very important for your patient, even me first, because I'm the survivor. Second, of course, Maurice Israel, and third one, Thomas Seyfried, agree on one thing: DHA and EPA is key. DHA, EPA.
EPA. Mm-hmm.
We used 500 milligram of DHA, 250 of EPA. Mm-hmm. And this what is doing this on the cancer cells? Thomas Seyfried, yes, it It's very good against ROS and against mTOR. Maurice Israel say it's blocked the keto paradox, and this way the ketones stay signalytic and not neutrons. But what we can see, it's the same treatment. If somebody is under a ketogenic diet and more the fifth essential, the 5 essential I just spoke about, you add DHA, and the cancer cannot use ketones.
Right.
And I can tell you that I have a French website with 9 million views, and we have a lot of people doing our protocol. Mm-hmm. And we have a lot of people who survive. Thomas Seyfried sent me, when people are they throw them from Johns Hopkins Hospital because it's too late. They can't do anything for them. Thomas Seyfried sent me some people, and I try to help them. I gave them what I did. And after, I don't know, because
they didn't tell you what's going on.
Yes, they don't call me again. And,
oh, that's yeah. So so what I'm getting from all this, Mr. Tenenbaum, is that you and your colleagues have done this extensive research looking at all of these different supplements, and the sense I'm getting is that the conclusions, despite some sort of local disagreements among you and Dr. Seyfried and Dr. Israel, is that there are likely to be some common mechanisms that are very inhibitory to these cancer cells, and that they may be coming from different interventions that are all sort of all the roads are sort of leading to Rome. Is that a somewhat accurate summary of where you're going with this? That with metabolism and nutrition, there are some key pathways that are being inhibited in cancer cells, and different dietary interventions may all be sort of converging on them. And the question is, can we figure out what those key sort of killing points are, and maybe figure out, what are the best doses, either nutraceutically or therapeutically, to sort of hit those points? Is that sort of a good summary of what you're saying?
Absolutely. That's why we have to do the Williamson dosage.
Yeah.
But we find the other way, the allopathic way. The allopathic way for us seems to, we can do a lot easier with medication, probably.
Right. Because you're hitting it on target more effectively.
And we know it will be uronefrex to inhibit OXCT1, an inhibitor of
mTOR,
inhibitor of cholas to inhibit ACAT1 and ACAT2.
Yeah. So I just want to ask you a question. So what you're saying, and here's Dr. Pizzolato connecting. I think, Mario sort of turned me into belief. I think if a year ago, we'd reached out, probably, I would have said, it's great to hear your story. I'm not sure we can help you. But today, after, looking at the patient data with Dr. Belusic, I think there's a ton of merit in what you're saying. So I'll tell you how my lab, we are approaching it. I look at large cohort studies, which, they have their positives and their negatives. The negatives are that, folks like yourself who, have responded exceptionally to sort of a tailored diet sometimes fall through those cracks, because, you can have statistics, but statistics don't take individual variation into account. But, basically, the idea is, one is, if you think about just prostate cancer, which you have managed to successfully beat for very long, metabolism in prostate cancer cells, and actually even just prostate cells, are actually quite different from other cells. So like if you look at a lung tumor cell or, colorectal tumor cell, how they use these metabolic pathways is different. So I mean, one of the things prostate cells do, and because, they, prostate basically produces, the nutrients to nourish sperm, right? So, they have this unusual feature where they just produce a ton of citrate. They don't use the citrate. You know this because this is what's going to go from acetyl coenzyme A into the Krebs cycle, right? So acetyl coenzyme A is sort of the gateway from, sugar metabolism, from fat metabolism, from protein metabolism. It's the intermediate, the carbon source that then goes into what, we call, aerobic respiration, burning calories through oxygen. And as you pointed out, cancer cells are very efficient at basically also using sugar. So this is what we call metabolic plasticity. So, they're very good at what we call aerobic glycolysis. Normal cells don't typically ferment sugars unless they don't have oxygen. Cancer cells can pick and choose. And so, so one of my caveats when I'm doing all of this is that I think, what you have said makes a lot of sense. But I the one point that I don't know if I would say disagree necessarily, but that I am wary of is this idea that there's a single gateway. I think one of the things that you've sort of touched on is cancer cells can use whatever they use. So, this idea of ketogenesis. I mean, I agree with both Dr. Israel and Dr. Friedman. I think depending on the context, depending on the cancer, depending on the stage, they may be able to use ketones. I think it just depends on what you're looking at. And I think this is, one of the challenges of doing research is how do you capture the context? And that's, one of the things Mario and I are trying to do with his metformin studies. We're like, okay, let us not look at prostate cancer as an aggregate. Let us just see what's happening in the hormone sensitive and then compare that to what's happening, in the hormone resistant. So again, my point is a few things come up, right? So in prostate cancer dietary studies, the ones that, if you talk to what they say, 9 out of 10 oncologists might tell you this, is that here are the things that, the studies seem to bear out. One is, yes, metformin definitely has some anti-cancer effects. You can argue about which stage it has the most effect. That's one. The other really big thing, right? So the other really big thing that everybody sort of agrees on is obesity, right? So if you obesogenic signaling, fat acts as an organ. Once get very fat, it acts like its own organ. And it's very inflammatory. And so inflammation is the one big thing. And this is not going to surprise you at all. And the inflammation comes down to, the balance between the pro-inflammatory and the anti-inflammatory cytokines. And why that makes a difference is, as Dr. Belusic said, part of that is an effect on the immune system. The other effect is on things like reactive oxygen species that we have studied extensively. I've studied this my entire career. And depending on which reactive oxygen species, they're not all the same. Some actually help cancer cells because they will drive signaling, mTOR signaling, for instance. And other related pathway that you might have heard of is this PI3 kinase AKT signaling. This is a survival signaling factor. But at some point, depending on, what else, like if you have very high iron levels in your cells, or very high copper levels, then you convert some of these signaling ROS into what we call damaging ROS. And these ROS don't help the cancer cell. They don't help anything. I mean, it depends on where they're produced. If they're produced in the cancer cell, they'll kill the cancer cells. If they're produced elsewhere, they will damage that tissue. So there, ROS doesn't care that where it's produced, what it does. But inflammation is a critical thing. And the other thing about inflammation is inflammation ties into cell death. And the inflammatory factor that ties into cell death is insulin growth factor 1. And I think you probably know this too. It promotes what's called apoptosis. It's a programmed cell death pathway. So, it's I look at metabolic charts and pathways. It's like a bowl of spaghetti. It's everything's linked to everything else. And cancer cells use all of those pathways to do what they need. But again, as you said, there are some key things. So the idea is to have an inflammatory profile that doesn't support the cancer cell, that supports the immune system and kills the cancer cells. And the second thing is, so for instance, fish, higher fish consumption correlates with lower tumors, likely to be because of, the changes in the inflammasome. The other thing is lycopene, cooked tomatoes. That's the other big one, right? And then the one that is negative, and Dr. Pizzolato and I talked about this last week, is dairy. There's a lot of things showing that dairy consumption. So there's this very interesting correlation. How tall are you, Mr. Tenenbaum, if you don't mind my asking?
It's very easy. Otto Warburg explained what is cancer.
Right. Yeah, it's glycolysis. They can switch to sugar when they have oxygen, and they shouldn't be able to, right?
Oh, okay. And after that, it's very easy to understand. Inflammation is the key, like you said. And how to not have inflammation?
Well, it depends on the kind, right? So because you can have the immune, the immunokines might help, right.
Omega-6, everybody knows, is inflammatory. Omega-3 is not inflammatory. So I eat, and it's why nutrition is very important. We don't eat sugar, of course. We don't eat inflammatory things. We eat non-inflammatory things. We don't eat dairy. We don't eat vegetable oil. And I almost finished. We don't eat supermarket meat because it's full of omega-6. And it's all a process. But where I'm not Maurice Israel told me it's very easy. They are only one case where cancer cell cannot build the membrane. I just told it. I said again for Dr. Pizzolato. It's OXCT1. It's inhibition of SCOT. Because like this, it's like, the door to go in a hotel.
Yeah.
Only in one side.
It revolves in one direction.
And we cannot go back.
Yeah.
So it's the only way for cancer cells to do the membrane and use acetyl-CoA. So it works for all cancer, because we have a result with on my 9 million viewers. We have a lot of people who prolong the life with our protocol, and it's a lot of different type of cancer. So,
would you be willing to consider the possibility that there may be something else? I was taught when you want to prove a hypothesis, you don't confirm it, you challenge it. So that's how we do everything. We believe in something, we do all these tests to disprove it first. And if nothing disproves it, that's how we know we're onto something. So, I really started looking into these SCOT inhibitors after John, connected me with your story. And I think it really fits with this idea that, tumor cells have unique metabolic strengths, but because they're greedy, they have unique metabolic vulnerabilities. I think that much is really clear. And they may be key points. I suspect there is more than one key point, because what prevents cancer treatment from being universally effective is redundancies and individual variations in how these pathways are activated or inhibited. So I guess what I will ask you then, Mr. Tenenbaum, is you're very invested in this, and you've really educated yourself. I wish my students would do the kind of due diligence you've done. But can you tell me a little bit of, like, let us say we partner with you, and again, metabolism and cancer, particularly prostate cancer, is now going forward the new focus in my lab. And as I said, we started by working with Mario on metformin. But as I told Mario, Mario has given us patient samples. we have analyzed blood and we've looked at the inflammasome, the cytokines, and then we are trying to mimic it in our models. And our models are what? So we have cell lines, and then we have preclinical. So we have, animal models. And then we have the idea that we might combine with other things. So what can we combine with to make it so that next time Mario runs this trial, it's not 40% of patients responding to metformin, it's 100%. Having said that, I will tell you, when we look at these patients and we group them into responders, non-responders, there's a lot of nuance and differences. So, we've pulled out we looked at 144 cytokines, and there's like 3 that seem to be common. And since we're recording, I don't want to completely divulge everything, but I will tell you, it definitely, there is something to be said for the fact that an inflammatory environment promotes cancer. And if we can hit that inflammation, then it definitely, seems to be correlated with those patients who did very well on metformin. So that's definitely there. Then the question is, what is key, and what is the key pathway that it comes down to? So it's a very for me, I don't think that there's a simple answer, but I think there may be a simple answer for specific cancers or specific patients. And so the goal for me is to work with, clinicians like Dr. Belusic, Dr. Pizzolato, and a rare opportunity to work with a patient like you to refine our model so we can do hypothesis testing, because something will come out of it. What I'm saying to you is that, if you were to tell me, Priya, we want you to show, SCOT inhibitors, work the best and killing cancer cells, how they work, then I would say, we can't do that. The first thing we would do is we would test whether in our hands this hypothesis, the hypothesis is that there is one route for cancer cells, and that route is through the SCOT enzymes. And if you inhibit that, you're done. That's it. You eradicate cancer. We will test that hypothesis, but the answer could be no. So then the question is, where we go from there? And my project, the project in my lab, is that we want to look more generally and look, rather than say this is this one inhibitor, one pathway that will work, we are trying to find a set of pathways, or like a sort of, not just one door, but maybe like a set of interconnected hotels, which if you block that street off, the cancer can't go anywhere. So I guess that's what I'm asking you, is that if we were to partner with you, and I'm certainly excited to explore that possibility, what would your expectations be? What would you like to see to make this worth your time? Because you're obviously talking to scientists all over the world. So, it's not like we're your only option.
Yeah. First of all, you're right, the cancer is multifactorial. But to heal cancer, it's multifactorial too. So each time we try something, we need to have this working fine. We have to do more this, like you said, ACAT1, ACAT2, we need it. When we inhibit SCOT, OXCT1, we need to inhibit thiolase for ACAT1, ACAT2. It works better this way. And that's why we have 5th essential. But what I will propose you is to first, what Maurice Israel told me, we have to do the Williamson dosage to know which one which one of all this siderophore and especially melatonin is the most powerful. And after that, I have everything and the way we have to do after. Maurice told me, don't say everything in one time, because you will lose your partner. And I spent almost 1 year with him explaining what happened. And so he told me, if you have question, I can give you his phone number and you can call him. It will be a lot more efficient than me. but the first things to do, it's because you asked me, it's the Williamson dosage because after that, when we will know which siderophore or if melatonin is the best way to go, you have even to do another things. You can compare melatonin with uronefrex. The uronefrex is the Spanish pimozid or Spanish litostar. And if melatonin is more powerful than uronefrex, we have the key. And they are more than 200 siderophores who are acid hydroxamic. So we have to find first which one is the best one. It's a very easy thing to do. Only by enzyme purify, Scott enzyme purify, and we need a spectrophotometer continuous.
So if I were to tell you, Mr. Tenenbaum, I would not approach it that way at all. I just wanted to tell you that because, if that's exactly what you want done, then, there are lots of labs that can help you, but probably...
Yes, but I don't find one.
What we would probably, want to do if we worked with you is I would go back and really delve into this, and we would start doing some preliminary, if melatonin is your interest, Scott, then we would do some preliminary testing in our cell lines. If the answer ends up being that in our hands, we don't see anything, my question is, what would happen then? Would you just be disappointed that this was where you invested and it didn't work? I mean, would we have credibility with you?
Okay, I tell you very clearly, we are sure it works. We are sure.
I'm a scientist. I only believe empirical studies, so I won't believe it till I do it in my own hands.
Yes, you know perhaps Brunings with a ketogenic diet. And this guy in Germany did, and he realized that people survive, but people survive only a short term. And after they die. Why? Because of the keto paradox.
Right. And that can do a lot of different things, right? You go on a ketogenic diet to lose weight, and I think Joe wants to say something.
Hey, Mr. Tenenbaum, always great to see you. So I think what Priya is saying is this is sort of an unprecedented thing to try to bridge these sorts of things that you're on and many people are on, and maybe not even necessarily on the same things, right? But how do we bridge your goals to getting into the more scientific avenues and proving, or even unfortunately disproving, some of these things? And as much as you're so deeply invested in Scott and in the metabolic process of prostate cancer, we have patients equally as invested in blue scorpion venom. And those things, and they swear that it works. And where the rubber meets the road, Mr. Tenenbaum, is when you have such strong beliefs, so strong that you're unwilling to test them because your beliefs are too strong to be willing to put that into the rigors of scientific testing. And I think what we're asking is really a little bit more leeway and saying, would you be willing to fund further research in the in evaluating metabolic aspects of cancer research, specifically prostate cancer, without I mean, I don't think we're, and while we can certainly be angled towards many of the things that you're interested in, I don't think Dr. Rai can really just say, we're gonna do only exactly what you want us to do, because I think we need to, there is a more of a scientific process, and this would be more of a gift to research saying we need to focus and study metabolic processes in prostate cancer. There is no question. I know Dr. Belusic is on the line, and he's one of my bosses, and so I would never say something that would jeopardize my reputation as a clinician, as a scientist, and as a person. There's no question that Mr. Tenenbaum has had a very dramatic response and prolonged survival to something that he is doing. And there's no question, and he and I, years ago, I even told him, Mr. Tenenbaum, I think you're a little bit too extreme, because he had, I don't remember the weight you had gone down to, but you must have lost well over 100 pounds. And you looked quite emaciated. And I was extremely concerned at that time. Nonetheless, your PSA disappeared. So, in all of that, I think what this whole meeting is really about is, would you be willing to fund a gift to the university looking
Absolutely.
at metabolism, specifically metabolism and prostate cancer? And, and I think more detailed than that, I don't think, it wouldn't be reasonable for that to be directed because of that scientific process. But with the goal being, hey, there are people who do better. We don't know if every prostate cancer patient does better with your diet. But we do know that, and I know on my personal, I don't know about Dr. Belusic, I personally put many patients on these extremely, for you, not so radical, 0 animal fat, 0 dairy, low calorie, keto-like. I don't want to say keto-like, because they do take in vegetables. And I definitely have a pile of people who seem to do better. Those patients are also highly motivated, right? It's not for everybody. Some people say, I'd rather live 5 years less and have my steak. And I respect that, right? We respect these patients, but this is sort of where we're at, because we understand the dramatic response that you've had, at least I do. And the question is, because of this, would you be willing to fund research looking directly at metabolism? Dr. Rai, if I got that wrong, please.
You have absolutely captured it. Again, Mr. Tenenbaum, here's what I will tell you. Let us say you do decide to go ahead and invest in this research. I will not be like, I forget, was it Johns Hopkins? I will be in touch with you. What I am looking to do is to get information like what you have shared with me and put it in context of the bigger picture that we're looking at, because I'm all about trying to fill in some of the blanks. I'll be honest with you. Metabolism. I'm a redox biologist, so I know a lot about ROS. And, you and I could have a long conversation about why I think in some cases epigallocatechin will work. Some cases it'll be absolutely terrible and you shouldn't take it. But, we won't get into all that now. We can later. If you want, we'll set up a Zoom call and, we'll chat about my training and what I think about, antioxidants and ROS. But exactly as Dr. Pizzolato said, this is, for me, this would be an ideal partnership where we get your input, we get your information, but, we do our projects, our science a specific way. I like to do these things holistically, and I will keep you in the loop, but we cannot do exactly the kind of protocol and the testing you do unless something else in our findings warrant that we do that exact protocol. And if that happens, I'll tell you. And if we test it doesn't work, I'll tell you that too. But yes, I think what Dr. Pizzolato said exactly captures what I was trying to say. So, then, I want to know what you think about that.
Yeah, I would just add one thing, that, we try to, we used to think that cancer is one disease, the prostate cancer is one disease. We know that every single patient has different type of prostate cancer. It's 100 different types of prostate cancer. When I was a trainee, when I was a fellow, there was a non-small cell lung cancer and small cell lung cancer. We had 2 recipes to treat those patients. Now we know it's 25 different diseases with 25 different pathways with different mutation. And we do precision medicine today, precision oncology, that we are trying to understand every single patient is unique. And having just one hypothesis that this is the key pathway in cancer works in every single prostate cancer patient is dangerous because you may really miss the bigger picture. And I think, doing systemic research and trying to understand the biology and understand that it's not one prostate cancer, there are 100 different subtypes, P10 mutation, MSH high, BRCA1, BRCA2, everybody's different. We're all different. Some people are tall, some people are this. Even people who are eating horrible diet don't develop everybody prostate cancer. So, there is something individual level.
A friend of mine called, doesn't matter, has a colon cancer. He made the same thing than us, except he ate a lot of meat, but only grass-fed and finished grass meat. And in 4 months, his cancer disappeared totally, like mine. So I don't know why, but don't think I have, I answer to Joseph, I'm not like this. I know that cancer is multifactorial. I know that we have a lot of way. And like I said before Joseph arrived, I said Maurice Israel said it's Scott and Thiolas. Thomas Seyfried said it's Ross and mTOR. But the treatment is the same, is DHA. So what I try to find, it's the best treatment. I'm not a biologist. It's your job to find why and what happened. It's not my job. My job today is to save people.
So let's do this, Mr. Tenenbaum. Let's do this. Let's fund research focusing on metabolism and prostate cancer. You'll be involved in understanding where that research is going. And, and people like Dr. Belusic and I really want to know who are people like you, because there are people just like you, but they're not all like you.
Absolutely.
And we want to know why. And Dr. Rye's research sort of gets towards that. That's where that goal is. Where are the Achilles heels of some of these cancers like yours? Your cancer has these Achilles heels. And there's no question, you're right. It may not be just prostates. There may be other people, but who are they and why? And that's what I think. I think this would be a good partnership if you funded that sort of research.
And yes. I will just say, Mr. Tenenbaum, I'm not actually specifically a prostate cancer biologist. So I'm sort of agnostic to the kind of cancers. I agree with what you're saying, what Dr. Pizzolato is saying. There are these 10, actually now there are 14 hallmarks of cancer, right? So every cancer cell has those hallmarks. The issue is there are very many different pathways that drive those hallmarks, and that may vary between patient and cancer and tissue type. But yeah, I think that is sort of that would be my ideal partnership, is investment in our research. We are working to get this funded through other means, but it's a new direction. And, all of these things, they take time and they take money and they take thought. And that's the fun part of my job. We would break this down into molecular level. Now, it may support some or all of what you just said to me, in which case you can tell me I told you so and now you've spent all this time. Or it may not. It may support some of it may support none of it, but it's going to lead to avenues that are exactly what you're telling me. It's going to lead to molecular interventions. And there may be molecular interventions, for a subset of patients or a larger subset of patients. But what I will tell you is I'm currently NIH funded to study repurposing a drug, an FDA-approved drug that had never been tested, in this case, prostate cancer, that we are putting in a clinical trial. It's a safe drug, and we think it's going to benefit metastatic castration-resistant patients. And I can give you other vignettes, from my research where we've done molecular holistic research, and that has led to a therapeutic, new avenue. So as Dr. Pizzolato said, that for me would be an ideal partnership.
I agree. I totally agree. to do this partnership? And are you able to do this dosage? The Williamson gave all the protocol to do this dosage, and it's very important to have it. If you cannot do it, do you know a laboratory who can do it?
So I'll be honest with you. I don't think that's what we'd start with. Now, in terms of finding someone, so, the way maybe to, if that's the idea, the agenda, then you may want to find, commercial labs, and I can look into that for you. So they may be labs.
I never find it.
There as well, let me see if I can reach out. I mean, there are labs here that do, nutraceutical research, and we can certainly connect you with some of those folks. I mean, they do what we call pay for play. You'll pay them, they'll do exactly, or very close to what you're saying. The way we do research in my lab is, I will tell you. So you've talked to Dr. Israel, Dr. Friedman. I think you talked to Dr. Berg. I believe you're talking to Dr. Sinclair, and you may have already realized this. Everybody has their own, favorite thing they look at, their own bias. That's the advantage. The academic research is very difficult. It's long hours. It's not all that well paid. But the one advantage is that all of us get to test our favorite ideas and collaborate with people and hear their ideas. And but that said, we would not undertake such customized research just because I would have to do my due diligence, and it would actually take quite a bit of time and effort from what we are already doing. And I have a small mid-size lab. I don't have a huge lab, but we can certainly look into that. But like I said, I think the ideas that you are interested in, and certainly you yourself are an example of, those are what interest me, and I want to be able to leverage those ideas into the bigger picture of what we're doing, with the hope that we will come up with a very defined molecular picture of what's happening, why some people respond, why some don't. The end result, maybe it may include some or all the factors that you've already done this research into, or it may include none of them, but it'll include something that's actionable. So, that's really our goal. So, but in terms of the specific testing, the Williamson dosage, those we can't do for you. But I mean, I'll look into who can, so that, you can have some hard data from these ideas of yours.
And I will give you, because you do allopathic treatment, I will give you something I hope that can be the same, but a lot easily. Scott, thiolase, and somatostatin. Try the three of them together.
Here's what I will do, Mr. Tenenbaum. I am going to, and trust me, I'm already doing this, and this is directly because of hearing your story. I will be doing my due diligence and looking into the research and all of that. And if I am convinced that there's enough rigor in that science, then I will absolutely translate that. But what I will just say is that I would not be comfortable with that being a precondition of any partnership. I want to be very honest with you, because I want you, in the end, if you invest with us, to be happy with what you're getting. I will look into all of those, but I cannot promise you that we will test that in the lab unless, I feel confident that there is merit in testing all of those. And I understand, you're yourself an exceptional responder. You're working with very well-respected scientists, but my research has shown me that a lot of established research is wrong, and it's wrong because it doesn't look contextually. We look contextually, and whatever the context we look at, we define that and we put that out there, with the caveat that it may not work everywhere. But with metabolism, there is a lot of nuance. And I'll tell you, the idea of your SCOT inhibitors, I could point you to like 20 studies that show that if you block, PDC, pyruvate dehydrogenase complex, which is basically what's, which goes through the acetyl coenzyme A, cancer cells will just pivot and they'll move to glycolysis. And when they do that, they enrich these cancer stem cells.
Can you send me these studies?
I will send you, and I can, if you like, what I can do is I can send you, some reviews which collate it, and then all the individual studies are in there, and I'm happy to do that.
Like this, I will send it to Maurice Israel for an answer for you.
Absolutely.
This guy is not stupid too.
Oh, no. Please.
I would like to tell you if you prove him is wrong, he will accept it. But
by the way, he may
when-
not be wrong. There's, my point is, I have found this context.
What you're saying. I understand what you're
I don't
saying.
want to say he's wrong or, anything like that.
Yes, I understand. I would like to tell you, we went step by step. When you see, if you know the studies of Brunings, you know that this guy lost all his patients after a while. And why? Because nobody spoke about somatostatin rise in 1970. So when you add to his protocol somatostatin, the guy survived. So, but they don't cure. And what we would like to say, like, there are a lot of people who said the cancer cannot be healed. You can survive a long time, but the cancer cannot disappear. But I think we find a way to cure cancer. That's it. I don't say it works for everybody. I don't say that. What I can say, it works for a lot of people who follow our protocol. That's it.
Yeah, I understand what you're saying.
And we have to find why.
Yeah. So I think we're up on the hour. How do you want to do this, Mr. Tenenbaum? I can discuss with Dr. Belusic, Dr. Pizzolato. But again, like I said, I think if this, and I'll certainly look for, any avenues for you to test, and I'll send you some literature. And I'll let you know if we feel that we cannot be the people to do that at all. I mean, like I said, I cannot let someone else direct my research based on someone else's research till I have done my due diligence. So, I think that's what you would need to think about. Having said that, we are going to be studying this idea of metabolism, metabolic syndrome in prostate cancer with the idea of extending it to other cancers. We actually have a collaboration with someone else looking at diet and fat and liver cancer. And like I said, we are all about bringing it down to the molecular mechanisms. That's what I'm interested in. Exactly, why do these things work? But I don't start, I test everything. I think scientists we're all doubting Thomases. We test everything. We're skeptics. But again, I test hypotheses by testing them. I challenge them. It takes us longer to publish papers and things than some of my colleagues. But I wouldn't do it any other way. Whatever we put out, we stand behind. So, that's where I stand. And certainly at the end of this, it's absolutely okay. First of all, it's a pleasure to meet you, and it's a pleasure to hear about what you're doing, because, God knows it gives us all hope. So,
I would like to help. That's it.
I understand.
The way you want, I will do the way you want. You are
perfect.
Then
we can discuss, I can discuss this, and then, maybe John will reach out to you and we can see where to go. But I'm going to see if I can help find someone who will do exactly what it is that you want to do. They'll ask you to pay for it, but maybe that might be a complementary way to go about it. And I'll send you some literature. Feel free to share, and we can follow up then, based on whatever is your comfort level. In the end, it's going to be your money, so you have, I don't like the idea that someone takes your money and then shuts you off, doesn't tell you anything. That's not how we would operate. But all I'm going to say is that if we do this, we may have information data that doesn't fit with some of these ideas. And so you don't strike me as someone who would be upset about that, but I just want to put that out there that, we find what we find.
I know that each professor, each Nobel Prize, I saw.
Oh, I'm not very far from that.
No, they have all of them another way, another belief. But my dream was to make them all together, to work together. But nobody wants. They want.
No, there's some practical issues. I think part of it is, I would, I know you're in Quebec right now. Is that right? You're not in Miami right now. By the way, I was in Quebec this summer. I thought it was an absolutely beautiful place. You're very lucky.
Yeah.
Well, when maybe you're next in Miami, I want to maybe bring you down and show you my lab, and you'll see the realities of why it's very difficult to put everybody together, and
with a lot of pleasure.
So
I will
yeah.
accept your
Yeah, we will bring you and show you, we'll take you down to the trenches. So, but in the meantime, we can discuss with John, and he'll reach out to you and and see how we go. But for us, it's either way. I think you've made your agenda very clear. Hopefully, we've explained how things would be done, and then you decide what you're comfortable with at that point.
And I tell you again, I'm sure I can ask all the professors I follow to help you. If you have questions, I will ask them to call you.
I'll reach out. I'll be in touch and absolutely let you know. And that's, I would appreciate if we can do something like that down the line. Yeah. So John, do you
No, Dr. Pizzolato, did you have something to add?
I just wanted to thank you, Mr. Tenenbaum, for taking the time and being supportive of what we're trying to accomplish.
It's always a pleasure to see you. I can call you Joseph?
Of course.
Call me Guy.
All right. So John is going to reach out to you, Mr. Tenenbaum, okay? And
okay.
This should be great. I appreciate it.
We'll all be in touch.
Yeah, we all absolutely. It was